Welcome to Ask the Expert. Today we have Bobbie-Jo Webb-Robertson, PhD, and Briggitte Frohnert, MD, PhD, to join us to discuss their work and their incredible new paper, Integration of Infant Metabolite, Genetic and Islet Autoimmunity Signatures to Predict Type 1 Diabetes by 6 years of age. This is really going to be an incredible new addition to the knowledge base. It came out in April 25th, 2022 in J Clinical Endocrinology Metabolism. And the paper highlights the power of collaborative effort in the type 1 diabetes research community, something Sugar Science champions. So let's just do a quick background. Dr. Webb-Robertson earned her PhD from Rensselaer. She specialized in the development of statistical models for protein homology modeling, and she joined PNNL in 2002, where she focused on development of methods for improving downstream analytics for mass spectrometry, divide proteomic, metabolomic, and lipidomic data for the purposes of improving biomarker discovery. So she's perfectly poised to enter the T1D realm here. In the past decade, she shifted her work to a machine learning driven feature extraction focused on biomarker discovery from complex and heterogeneous data. In the field of type 1 diabetes, she's working on machine learning discovery efforts for multiple birth cohorts, such as TEDDY, the environmental determinants of diabetes in the young, and DAISY, the diabetes autoimmunity study in the young, and as well as helping to design and guide the data science needs for the network for pancreatic organ donors and diabetes, which is NPOD. Her full publication list can be found in Google Scholar. Welcome. And Dr. Briggitte Frohnert, MD, PhD, she's an associate professor at Ann Schutz Campus School of Medicine in Colorado. And her recent interest is understanding the environmental factors which contribute to development of islet autoimmunity and progression to type 1 diabetes, with the ultimate goal of preventing type 1 diabetes in the future. In 2014, she joined investigators on 2 large epidemiological studies, DAISY, which I alluded to before, and TEDDY. And TEDDY actually followed over 8,500 children. DAISY looked at 2,500 high-risk children. And with a background in molecular biology and biochemistry, she's interested in the use and integration of omics data, which omics is considered genomics, proteomics, metabolomics, et cetera, to generate new insights in the disease process. So welcome, Drs. Webb-Robertson and Frohnert, and I'm very interested in hearing about your work.
All right, I will go ahead and share our presentation here. Can you guys all see the presentation?
Yes, you might want to click. Yeah. Oh, that looks beautiful.
Okay, so let's make sure it wasn't in the notes format.
Mm-hmm.
So thank you very much for that introduction. So I will not reintroduce myself or Briggs, my co-presenter here. I did want to start with the sort of acknowledgement slide without reading everybody's name. This work really has been a very large multi-institutional and interdisciplinary collaboration, including the Barbara Davis Center in Colorado, University of South Florida, Virginia, Helmholtz, and PNNL, or ICIT. A lot of this work was funded by TEDDY and the NIH. And here's the specific link to the paper we're going to talk about today. But I actually thought what I would do is start with an introduction to sort of the field of artificial intelligence, because we do get a lot of questions about, what is AI, what is ML, what is deep learning, all of these things. So I like to put it in this context. AI is really the really big field that includes everything, not just the algorithms to make prediction, but the robotics to build, the sensors and the robotics and all the pieces that actually make an AI function. Machine learning are those actual learning algorithms, and they can be anything from simple rule-based algorithms to statistical learning methods, which is what we actually employ in this talk. And then deep learning is actually a subset of machine learning, works very well in certain situations. AlphaFold, for those on the call who are familiar with AlphaFold, really put deep learning in the bioinformatics super category, but it works in very specific scenarios, but it's built on very deep neural nets. For the statistical learning methods that we use, there's some strengths, but there's also some challenges. The strengths are the multidimensional data that they can manage, looking at multivariate signatures, but there's also some challenge that it requires labeled data. In many cases, a lot of labeled data, which can make interpretation sometimes hard if you're doing it with small amounts of data. Overtraining, so you can get propagation of error, so you could train an algorithm, and if it's actually picking up the wrong things, as you add more things, it can continue to do that and propagate that error. Then depending on the method, it can take a lot of computational resources or be hard to interpret. So, we got to always we pick the best method that we think for the problem we have in hand. And sometimes that changes over time, but that's really the category of work that we're talking about here in terms of machine learning.
And can I just quickly ask, how old is AI ML, that sort of application?
So
in
AI
the healthcare field?
Yeah, it's actually been used for decades, right? Early on, there were a lot of expert-based systems that were built that were rule-based, and they still were considered machine learning, right? If patient A has blood pressure over this value and this marker over this value, then prescribe this, right? That's still making a prediction of what's the right treatment for that patient, just based on simple rules. So it's been used for decades, but in the sort of this statistical learning framework, not quite so long, at least for type 1 diabetes. So I actually went to PubMed and I did the most basic of searches, machine learning, type 1 diabetes. And that's what this graph is showing, is that, back in 2015, there were like maybe 3 papers published that included both of those terms.
Yeah.
And in 2021, it was like 73. So that's a huge dramatic increase. Yes. And it's much more than that, right? Because a lot of people don't put the term machine learning in their title, but are still doing that as part of their paper. So I think you can really see the dramatic growth in really the last 3 to 4 years of the application of machine learning in the field of type 1 diabetes. I'm not going to talk about these 3 applications, but what I wanted to show by just putting these 3 up there, and you can look at the titles, is that the ways people are using it is very diverse. Everything from image analysis to biomarkers, which is what we do, but for different technologies such as single cell, or looking more at clinical measurements like glucose monitoring and trying to predict what's the next measurement that's going to come up for this patient so you can get ahead of specific issues. So it's being used very heavily right now in the field of type 1 diabetes and in a lot of different application spaces. So what I'm going to talk about today is just one of those application spaces. But I think this does show that, there, despite the caveats to machine learning, it's come a long way and it can do a lot of good.
Yeah, no, it certainly is getting more popular, and that's great because as more people use these tools, they can, go back into, even as we're going to see, go back into pre-existing data sets and really pull new things out.
I agree. So here's an example. Neither of my 2 titles would be in that PubMed search because neither of them use the term machine learning. But I did want to point out, we're talking about this paper down here, and I don't have my laser pointer on, but I can put it on. We're talking about this paper down here, the integration of infant metabolite genetic and islet autoimmunity signatures for type 1 diabetes. But we've had 2 prior publications looking at different endpoints, but with a similar theme. So what our sort of theme in machine learning has been in this sort of collective group working together has been, how do we sort of integrate traditional risk and genetic biomarkers, which are actually a little bit simpler because they don't change dynamically over time, with dynamic functional biomarkers like proteins and metabolites to improve the prediction of specific endpoints of type 1 diabetes. Before we get into sort of the results, I want to point out one thing about biomarkers is they can be measured in lots of different levels. And when we do a lot of the biomarker work that we talk about in our paper, they go all the way from discovery phase to validation phase. But most of our work is way down here in the discovery phase, where you're measuring as many things as you possibly can, hundreds or thousands of proteins, metabolites, And then you're trying to mine that to find what are your best candidates to move on to verification. We have verification data now in TEDI, although it's not presented in this paper. And then ultimately the goal is to validate this, right? So that you have something you can actually put on an assay and help people with. But we're still way down here. So we're trying to use machine learning to learn something about the patients and what might actually be useful in the clinic as predictors. And I'm not going to go into a lot of detail, but this is sort of our basic approach. We don't put all data together into one specific machine learning algorithm. We let the data choose, set up some sort of fusion, then feature selection. Then the evaluation piece is important because for our particular application, we're not only interested in accuracy, we're interested in what features are driving the prediction, so that then maybe we can move on to those other phases of the biomarker identification to build more predictive models that will work in a larger population.
What's the timeline for that process?
So for TEDI, for example, going from discovery to verification data was like 5 years, but it was an extremely large number of samples. Yeah. But it can be done in a few years, usually, if you have really dedicated resources.
TEDI was 8,500 children, right?
Yeah, we didn't analyze all 8,500, but we did analyze a lot of samples with, for example, proteomics. But every data type for TEDI is at a different phase. And so I'm giving you that timeline for proteomics. It's shorter for metabolomics.
Yeah.
So it depends on what you're measuring.
Right.
So for TEDI, for this specific prediction that we were doing in this paper, will the child progress to type 1 diabetes by age 6? We had 73 kids that had and 582 who did not. And these kids, this is sort of the flow chart going from the full set of TEDI. How do we get down to 655? We wanted complete data for HLA, autoantibodies, dietary, growth information, GRS, as well as metabolomics at these 3 early age time points, 3, 6, and 9 months. Because we wanted to know, could we predict it from, the measurements we have on these kids when they're very, very young. And we used the method I referred to before, this repeated optimization for feature interpretation approach. And when you run machine learning, I added this to sort of give you a sense of what happens is you put in all this data and you crunch it through your algorithm, but then for every child, you get an individual prediction. So, these are my T1D. It may say, we believe that this child will have T1D by the age of 6, given their information, with a probability of 0.97 or 0.82. And this might be a false positive, right? 0.71. So you don't only just get a yes, no, you get a degree of how likely, the likelihood that a child will progress to whatever endpoint you put into the machine learning, given the data that you have for that child. And this is where I'm going to hand it over to Briggs to talk about the results. And Briggs, just tell me next slide, and we'll go from there.
Great. So here on the right, we have the features that were selected in the analysis that Bobbie-Jo just described. And for each, the bars along the right represent feature importance. So basically in the iterative process of selecting features that predict the outcome, the higher the number for feature importance basically tells you the number of times that that feature was selected in the algorithm. So for instance, at the very top, in the very top, so if you can click once, Bobbie-Jo, the arrows will pop up.
Oh, sorry.
Oh, there we go. Cool. So these are in the brown bars are features that were selected at least 50% or more of the time. And so in that top cohort of the most highly selected features, you see the very number 1 is islet autoantibody positive. So basically at 9 months, if you have a positive islet autoantibody, you are highly likely to go on to develop type 1 diabetes. And that is not surprising from what we know. Some other highly selected are features that we also know from established work that are high risk factors, such as the HLA DR3/4 genotype, which is the highest risk HLA type. And then the next one right underneath it is GRS. So that's the genetic risk score. So that is a risk score that has been developed in other populations and refined in TEDdy, looking at multiple SNPs, including HLA, and using those to generate a number that gives you a prediction of likelihood of developing type 1. We also have cow's milk formula and gestational age. So those are previously identified risk factors. And then you press the button again. So the starred factors are factors that either these metabolites or very close related metabolite showed up on the DAISY analysis that Bobbie-Jo showed a few slides ago. In the DAISY analysis, we looked at kids who, we had 3 populations: controls, kids who developed islet autoimmunity, and then kids who progressed to type 1. So there was 2 analyses in that study in terms of selecting features. One is risk of developing islet autoimmunity, and the second was from those who developed islet autoimmunity who developed type 1. And most of these features selected come from the islet autoimmunity, with the exception of I think valine, which is down kind of about 2/3 down the chart. And then if you click the button again, so the yellow dots represent, these are metabolites that were selected in a previous study in TEDdy that looked at longitudinal metabolomic profiles and selected metabolomic features that predicted outcome of islet autoimmunity. So I just wanted to stop a second here and kind of clarify. So this analysis is looking at outcome of type 1. DAISY looked at both autoimmunity and type 1, and then the TEDdy metabolomics analysis was looking at endpoint of islet autoimmunity. So a little bit different endpoints. And one of the interesting things is you might have different features that select for one part of the progression versus the other. And so a couple things I just wanted to point out. So several metabolites could be dietary based, so piperidone and ascorbic acid. So ascorbic acid is, I think, about the 8th from the bottom, that showed up in both TEDdy and metabolomics, as well as DAISY. Vitamin C is known to inhibit insulin secretion in mice. And so increased level at the earliest time points is associated with islet autoimmunity. In both TEDdy and
sorry, just with ascorbic acid, like how much over the limit is that?
Okay, so what
or
this number
over
is,
the normal.
What this number is telling us is that continuous variable, when put into the machine learning algorithm, will be selected about 20, it looks like 22, 23% of the time as a important feature predicting outcome. So, and Bobbie-Jo can jump in on this, but one of the important things, it's very tempting to look at the list and then start thinking about the individual components.
Right.
But what this really is telling you is that these are ensemble features. And so to predict, you have them together in the algorithm. So if you are then going to go back and look at just an individual feature, it's a little bit different, important question in terms of validation, but it is a different analysis then. Bobbie-Jo, do you want to add to that?
I totally get. Yeah, that's very important clarification. It's like it has to be seen, like you said, as an ensemble.
Yeah. And of course, the higher it is on the list, the more frequently it was part of that ensemble. So I think one of the points of kind of indicating these crossover features is just, I think that this is encouraging to see that there's crossover between different outcomes and different studies. Well, similar outcomes, but different studies in terms of what gets selected. The other thing I wanted to highlight were both TEDdy and DAISY selected metabolites in the branch chain amino acid pathway. So those branch chain amino acids are leucine, isoleucine, and valine. So valine was one of the ones that picked. I'm trying to remember where that is on the.
There's valine.
There it is. Valine
and isoleucine.
And isoleucine. So the branched chain amino acids are intriguing. They are also selected in metabolomic studies of type 2. Branched chain amino acids are known to potentiate glucose-stimulated insulin secretion. So it's sort of a cross disease signal. And then some other features that got selected here were sugar metabolites, so fructose, levoglucosan, glycerol alpha phosphate, and xylulose. God, I can't say the word. So suggesting that kids who develop type 1 might have differences in their carbohydrate metabolism. Interestingly, in the DAISY analysis, we picked up sugars in the islet autoantibody to T1D progression analysis, but not in the start to islet autoimmunity outcome. So, and this, of course, this analysis is going to pick up both sort of steps in that progression. And then another thing that came up in all three analyses was the gamma aminobutyric acid, or GABA, which is known to be important in regulation of secretory function in beta cells, or at least in vitro. Also, GABA is produced by GAD65, which is a target of autoimmunity. So some interesting common themes between different studies. So
it's very interesting that you've captured this snapshot of like from early days. It looks like things are not quite right.
Mm-hmm.
Yeah, it's a very good point. So both the TEDdy metabolomics analysis and the DAISY analysis are looking at kids who of a broader spectrum of age, but this is really looking at those early time points, which is very interesting. So next slide. So moving away from looking at the individual features, now looking at how useful is this ensemble in terms of a screening tool. So, so basically along the y-axis, we have the area under the curve, and along the x-axis, we have different scenarios. So in the first scenario, none, that is basically what is the area under curve for prediction of T1D outcome if you use just the data that doesn't come from a blood draw. So that is growth data, demographic data. And so you see that's kind of your baseline. It's not a very great prediction, but indeed there's some prediction there. Then the next set of box plots are if you're basing your screening on only the 3-month blood draw, but you're screening at for the reddish brown bar is 3 months, the black bar is 6 months, and the golden bar is 9 months. So if the screening happens at those different ages, but you only have a blood draw from 3 months, that's your area under the curve. At 6 months, it's really, if you're just looking at the 6-month time point, it's not much different. So a little bit, not significantly different. But if you look at the 9-month blood draw, now suddenly your AUC goes up quite a bit. And of note, that is the first age where we have islet autoantibody measurements. And so as you recall from the feature selection, that was the most highly selected feature. So not surprising, that piece of information really ramps up your predictive value. And then the last groups are basically if you use more than one blood draw information. So 3 and 6 months, 3 and 9 months, 6 and 9 months are all 3 time points. And I think kind of the important thing here is that 3 and 9 months seems to be significantly better than just the 9-month, whereas, and not all that different from 3, 6, and 9. So basically that 6-month time point is not adding a whole lot in terms of your predictive value. Next slide. And so what this is looking at now is in the top set of graphs, we're looking at the box plot of the AUC, looking at using different data sets. So we have the islet autoantibody information, we have the genetic risk score, the GRS, we have MET, that's metabolomics data, and then we have HLA type alone. And keeping in mind that HLA is a component of GRS. So if you are looking at only one single data set, the best AUC comes from islet autoantibody alone. In the next group, if you look at combinations of data sets, you can see that the best prediction comes from a combination of islet autoantibody and the genetic risk score. And then when we do 3 data sets, addition of HLA and genetic risk score, or metabolomics and genetic risk score, plus HLA are pretty similar in terms of prediction. And then finally, if you use all 4 data sets. So, and then in the bottom set of panels, what we're looking at is if you restrict if you restrict to only cases where you have a low false positive rate, so false positive rate under 5%, your true positive rate, how is that impacted by using the different sets of data? So when you restrict to a low false positive rate, so you're trying not to pick up kids who are not truly going to progress, now the metabolomics look more important. So basically, if you look at the single data set, pretty similar pattern to what we saw when we did the whole spectrum AUC. But now when we look at the combinations, the islet autoantibody plus metabolomics performs best. And then finally in the 3, metabolomics is in the combination of 3 data sets that works, that gives the best true positive rate. And then all 4 data sets is really not that much different. But one thing to point out is if you look across the board, the best true positive rates are still in the 30s, right? So it's not a super high true positive rate. Next slide. So just to kind of summarize here, oops, and just in time. So the prediction is relatively high in terms of the area under the curve of 0.82 with our best prediction. But our true positive rate was not high enough for practical use at a low false positive rate. Validation studies would be needed so we can further refine. The other thing we have is that the optimal screening age was 9 months, where we now can incorporate that islet autoantibody data. And it does increase significantly if you add the 3-month data point for additional information. And then finally, feature importance for metabolomics can help in identifying important pathways. We kind of highlighted some of those that pop out in multiple studies. And in this group of kids who progress by 6 years of age. And finally, the feature importance tells you that the genetic risk score and HLA might have partially duplicative information, which we know since the GRS includes HLA plus other SNPs. And with that, would welcome further questions.
Yeah, that's great. I mean, this is just great. It really, I mean, you said it took 5 years, right, to kind of wade through these massive data sets, but really you were able to pull out some very valuable pieces of information. And I'm going to open up to questions. I wondered if I have 3 questions. One is, how much more value do you both think that MLAI can add to the T1D clinical study landscape? In terms of going back over like even papers and studies and pulling out new insights. I mean, do you feel like there's a lot more to go? Have you feel like you've captured a lot of it? What's your thought?
You want to take that first, Briggitte?
I can take that first. So one of the, I think there's room for going back. I mean, we have these studies like DAISY, Baby Diab, DIP, DIPUS that have been going on for decades. So a deeper data set in terms of length of time, but perhaps not as robust data collection as in TEDdy. And then you have huge amounts of data from TrialNet. So I think there's a lot of data there that has not yet been looked at or is just starting to be looked at using some of these methods. And I think I'm working with a collaborative called the Tie-Dye Collaborative. So that's DIP, DIPUS, DAISY, Baby Diab, and DO-IT. And harmonizing our data and then using AI or machine learning to pull out things like antibody patterns or trajectories where you combine age of an antibody pattern and the constellation antibody pattern. And I think there's some interesting insights you can come out with that. And then what's really exciting is when we get more richer genetic data, can we overlay that with things like antibody pattern, and can we start to discover not just overall risk genes for type 1, but risk genes for specific pathways. And to some degree, you can do that by looking at just one feature stream, but I think combining things is really interesting to see if there's, particularly if we're trying to get to this customized, individualized medicine, where we're trying to target interventions to people with specific pathogenic pathways.
Yeah, I mean, and it makes perfect sense, right? If you get this layered approach going, you'll have much more informative data and sort of you'll be able to dissect the prodrome and then sort of attack it clinically, personally. And I mean, people say, oh, well, you don't want to sort of cut things up into endotypes, but I mean, that's exactly what the breast cancer field did, didn't they? They had, it was just like one size fits all, and now it's very customized, which, could be really a win for the type 1 diabetes community too, if you could sort of cut off progression at certain time points. I wondered, what's the timeline, just sort of like a ballpark timeline for pinning down the appearance of autoantibodies in those at risk and getting these diagnostics into the newborn nursery, or like at the 9-month, right, pediatric well child checkups? And what are some of the roadblocks?
Okay, so right now in the US and Europe, there's some momentum behind designing screening programs, and particularly as we have our first intervention hopefully coming online later this year, that becomes even more a more convincing strategy.
Yes.
And so Germany has looked at one time point, now two, looking at islet autoantibodies. We are looking at islet autoantibodies. Virginia is looking at first screen by genetic risk score and then following up with islet autoantibodies. South Dakota has an interesting, I think, combination where they're doing newborn screen for genetic risk score and islet autoantibodies in all children. So I think that's going to be helpful, because one of the things is that, and then cascade. So Washington State is doing genetic risk score followed by islet autoantibodies. So I think those different studies are going to be helpful, and hopefully in the next 3 to 5 years, more information in terms of how do we do in terms of ability to identify kids. We know that, Teddy, Daisy, et cetera, that we deliberately were selecting high-risk kids to begin with and then looking at them, but we know that that group does not represent all new onset. And so these screening studies are going to be really important. So I'm hoping that once teplizumab hopefully gets approved, that will light a fire under some of the foot dragging around, like, why are you screening for something you can't do anything about? So I think in the next 5 years, we're going to see changes. I don't know if we'll be able to go universal. One of the big things we need is American Academy of Pediatrics, Bright Futures, to get on board and say, this is worthwhile, we should be doing this.
Yeah.
So it gets paid for.
I mean, even pushing off diagnosis, especially at the young age, by months to a year or so is significant. And also just identifying those, that are risky, they can be monitored more closely and gain more information from that particular patient.
Yeah, there's 3 advantages. One is, can you prevent somebody from presenting in DKA? And we know that our studies can do that by monitoring. Right. The second is, can you, and we also know from analysis that we've done that has, and we and others have shown that DKA at diagnosis has impact on long-term A1C trajectory, which has impact on long-term complications, which has real sort of financial benefit in terms of our healthcare system as a whole. Absolutely. In addition to the benefit to the people who aren't having their kids in the ICU. And the second is the potential to intervene and potentially over the coming years, either slow or completely stop progression. And then the third,
almost like in a remission status.
Yes, and sort of like we do for other autoimmune diseases, that we try to keep things as quiet as possible for as long as possible. Yes. And then the final is, I think, giving families tools so there's the advantage of not being in DKA, but even so, I think for people to know that this process is coming and that insulin is in their future is less stressful than one day you're in the ER, you find out your kid needs insulin for the lifetime, and it doesn't matter what you had planned for this week. This is now what you are doing, is figuring out how to manage this crisis. And so I think there's multiple layers at which it's a benefit to the community.
Yeah, that's great. I mean, I know you're in the clinic also, so that that's a great perspective at sort of to hear. I guess my last question is, what is your thoughts about sort of this whole, there's a non-coding genomic element to the progression or even the existence of type 1 diabetes. How might ML and AI be used to address that or, kind of get at that?
Do you want to try that one, Bobbie-Jo? I mean, that's kind of a lot of what we've done is focus on identified SNPs, right? So that would be, I think, harder because it's a
Maybe
bigger.
Kendra. She has some questions too.
Okay.
Yes, Kendra, please join us.
Unmute. I'm unmuting myself. Hi, guys.
Hi.
I have a couple of questions. And while I was typing that, I wasn't listening to your question, Monica, so I'm sorry.
Oh, I just was saying, how can ML and AI, is there potential for it to further dive into the non-coding genes that maybe have been implicated in type 1 diabetes?
I don't think I can answer that question as well.
Need Steve Rich on the call.
I know we've talked about using Bobbie-Jo's approaches to whole genome sequencing data, and that there is a computational limit to how many things you can toss in the pot and get to an outcome. Also, we're limited by number of people and number of data elements.
Okay, to be continued on that one. But so Kendra, what did you want to offer? And
well,
congratulations, you're on this study as well.
Bobbie-Jo and Briggs, really nice. And my two questions, the first one is to Briggs. I think you did a nice job trying to tell us about what kind of pathways and the types of metabolites that were used in prediction. And I was curious, how similar are those, or is there ones in particular that are similar to what we see in IA, what you guys found in IA with your previous papers?
Yeah, so the IA pathways that that seem to pop out are, so there's the branch chain amino acids, the ascorbic acid came out in our IA endpoint, and also in the general TEDdy longitudinal metabolomics. As I mentioned, like the sugar kind of pathways didn't come out in the TEDdy analysis because they were looking at IA as an endpoint, but they did come out in our IA progression to T1D. So I think that's a whole other, I mean, there's kind of, it's a continuum, but it is interesting to look at what happens early before IA, what happens later between IA and diagnosis. And most of the other pathways that got pulled out were actually pulled out from the in the endpoint of IA. So like the pentose phosphate pathway, which I didn't mention, but is also pulled out from multiple studies. GABA, those all came up with IA outcome. And really the biggest thing that came out in our panel of outcome to T1D was sugar pathways. So maybe less exciting from immunological point of view.
Well, no, and I think all of it plays out together. And like you said, there's two different pathways. And how do you, I guess, in a broader, take this predictive type of modeling and move it into a causative plane? How do we start to really understand it mechanistically, or use this information that we get for prediction and use it in more mechanistic studies?
That's such a good question.
It is a good question, and therefore does not have an easy answer. I mean, I think, one thing so our approach thus far has been, I mean, I think with TEDdy, we'd see this a lot. Do we look at an individual food group, or do we look at a food group pattern? Do we look at an individual metabolite, or do we look at a pathway? And there's kind of benefit to both angles. I think validation would be great. I mean, we have now a set of pathways that are of interest, and we're starting to have all these kids from the general population screening. What if we started looking at, some of these ensembles in kids and look to see, like, what does that say about time to, although most of those kids are coming in at islet autoantibody positivity. So maybe that's not the greatest situation. It is a little hard. Like if we took 3-month-olds and we screened them for genetic risk score, but we also looked at a panel of metabolites. I mean, one of the things that Bobbie-Jo mentioned was that the true positive rate was not that high. So it's of limited use to screen a population. How do you improve that, Bobbie-Jo? Do you think like if you go back with another data set and you refine to a smaller number of metabolites, or you limit yourself to specific pathways, would that help?
It might. I mean, so to Kendra's question about causal modeling, that takes massive amounts of data to do it computationally. And part of the issue, even this paper and all the other ones, right? It's only 73 of our kids had progressed by that age. So that's a small population. So you have to do those validation studies and maybe tweak. So there's lots of ways to do it. It's all just time and money, right? So right now we're just saying how well are we doing globally, but you can modify your optimization metric to focus on just true positive. If you have a false positive rate that you're most interested, if I could hit this, then it would be clinically useful. And that's really what we should be optimizing for, not this general overall area under the curve. We should only be optimizing to that specific goal or target. So that would be one thing we could do. We might find more useful predictors doing that. It's probably something we should do moving forward.
Thank you. Thank you.
Yeah, and I kind of wonder, it depends on what we're doing with the information. So if the information is having us focus on these children more closely and have them come back for a follow-up visit, another lab draw, further measurement of antibodies, that's pretty low risk. And so you might tolerate a pretty high false positive rate, right? But whereas if we're selecting people to put into an intervention study, that's going to be different. So that's the other piece, I think.
But you still have to pick. So if you wanted to get all your T1D kids in the paper we just presented, right? And we have 500 and some negatives and 70-ish positives, you would have to then move forward almost like 400 of your negatives to make sure you got all your positives. So is that too many? Maybe not, but I think so, right? So I still think you need to pick it. You need to pick what is reasonable, right? So you're like, sure, I'd be willing to do a 1-to-1, right? If only half the kids that I move forward actually truly do progress, I'm happy with that, because the cost is low to screen the other half that will be negative. Then, but machine learning, that's always a challenge, right? You got to be optimizing to the right thing, and the field is, in general, when people, just data scientists come in, the first thing we optimize to is AUC. But now I think we've learned that maybe through practice that it's not that simple. And maybe we need to get more focused.
Is there a paradigm? Is there a paradigm for this in other disease states that could be useful to the type 1 diabetes research focus?
It's a good question.
Like certain types of cancer research, or
there. I'm sorry to say I'm not familiar with.
Is anybody else doing this? I mean, is anybody else mining for these?
They're starting to open up causal mechanistic methods in cancer. I am seeing a lot more of that, and I'm seeing a lot more a priori, like they're really targeting one metabolite in particular, and they're looking at how it's related to the immune system. And another area that I've seen it in is aging. Yeah. So specific metabolites, and they've done some clinical trials in that area in aging, and and they're working on developing supplements that increase those metabolites, and also seeing very good results in their interventions. So I do believe there is some out there. I just from all the work I've looked at, it just seems like it's really limited it's limited to number one prediction. And I think that both Bobbie-Jo and Briggs have done a lot of work in this area, specifically because they have access to great data sets that have that information. But the other thing is, I think it's going to be hard to validate because of that, because we do have really granular measures over time, where that's hard to capture in other studies. So, you know, it comes down to money really in developing those big studies.
Yeah, I agree.
Yeah.
Okay.
Yeah, I mean, I think,
Do you hear that, NIH?
yeah.
Sorry if I cut you off. What were you
Oh,
gonna
no,
say?
I was just gonna say, I think also be interesting. So we're doing this as a single cross-sectional, but I think would also, well, no, that's not true. We had multiple, but I think it'd be interesting to see if you had a two-step selection. So first you select people on genetic risk score, and now you look at the other features. Would that work better? So if we went to a screening model like the South Dakota Stanford Health group is doing for general population, where you first do genetic screening, and then you look at everybody at age 5, I think it is, for antibodies, we would be able to potentially do, you know, that early screening and get some extra data, extra blood, look at some metabolites, maybe, and get a sense of how well sort of selecting a population helps you refine who you're gonna. Does that improve your true positive, false positive?
It seems like, I mean, it just seems like, just from a lay person's, you know, view, it seems like it would.
Yeah, so I mean, I think that's the one place where we could get more data, because Kendra's right. I mean, the Teddy study is not going to be duplicated to the way that, I mean, the amount of time, effort, I can't believe what these families have done in terms of samples that they've provided. It's pretty massive. So,
Yeah. I mean, the thing is, I think once people are in it with the diagnosis, they're quite hopeful and desperate for, you know, a change in the way that the disease has to be monitored and treated, and, you know, like, you know, in their wishes of wish, they have the idea for a cure. But it is this is I think a really sort of a great new paper. You know, you're diving back into your previous work and kind of like bringing it together, and I think it's really setting the stage for just more of more inquiry, and the great way the collaboration was put together. I mean, it was just really exemplary on many fronts. So thank you all for, you know, getting this work out there and published, and hoping that you'll, you know, have more insight soon.